Hello, hello everyone. Welcome to lecture number 2 in our comprehensive series on the chemistry of carbohydrates. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. In lecture 1, we established the definition, classification, functions, and isomerism of carbohydrates. Today, we move from open chain structures to the biologically relevant cyclic forms, understanding structures, anomers, and mutarotation is absolutely essential because over 99% of the glucose in our body and tissue exists in cyclic forms, and enzymes, transporters, and receptors recognize only these specific conformations. Let us proceed systematically. So let's discuss the structural representation of sugars. Three major ways are used to depict sugar structures, each serving a distinct process. The very first one is the Fischer projection, that is straight chain linear representation, ideal for showing dextro or levo configuration, and open chain aldoses or ketoses. Second one is the Haworth projection, simple perspective view of the ring form that shows the relative position of the hydroxyl groups and the anomeric carbon. Number 3, that is conformational representation, that is chair and board configuration. Most accurate three-dimensional depiction reflecting the actual spatial arrangement due to the tetrahedral geometry of the carbon atom. In clinical biochemistry and molecular modeling, we frequently switch between these representations. Chair conformation is particularly important because it explains steric hindrance and stability of different anomers. So this structure showing the pyranose and the furanose ring structure, right? Monosaccharide cyclize to form the rings resembling either pyrene and the 6-membered ring or a furanose or furane, that is 5-membered ring. Let's talk about the pyranose, 6-membered ring, the glucopyranose. In aqueous solution, more than 99% of the glucose exists in pyranose form because it is thermodynamically more stable. How the furanose, 5-membered ring, the fructofuranose or ribofuranose in RNA. This ring nomenclature is derived from the heterocyclic compounds pyrene and furane. In human metabolism, glucose is transported and phosphorylated almost exclusively as the pyranose form, while ribose in nucleotides predominantly adopts the furanose form. Understanding this helps us appreciate why certain sugars are preferred in different biological contexts. This shows the chemical structures of furane and pyrene rings. Furane is a 5-membered aromatic ring with 1 oxygen, while pyrene is a 6-membered ring with 1 oxygen. When sugar cyclize, they form non-aromatic, saturated version of these rings. The oxygen atom in the ring comes from the hydroxyl group that attack the carbonyl carbon. This structural analog is useful for remembering ring size and stability. Let's talk about the aldose ketose isomerism. This is just basically the recap for the emphasis, right? Fructose, the ketohexose, or the glucose, that is aldohexose, share the molecular formula, that is C6H12O6, but differ in the position of the carbonyl group. In glucose, the aldehyde is at carbon number 1. In fructose, the ketone is at the carbon number 2, the anomeric carbon. This difference profoundly affects their cyclization pattern and metabolic pathways. In the liver, fructose bypasses the main regulatory step, the glycolysis. That is phosphofructokinase 1, which has important implications in conditions like fructose intolerance and non-alcoholic fatty liver disease. Now, let's talk about the alpha and beta anomers. When an open chain aldose cyclizes the carbonyl group, carbonyl carbon, the C1 becomes a new asymmetric center called the anomeric carbon. The two stereoisomeric or stereoisomers forms are known as anomers. What about the alpha anomers? The hydroxyl group at the anomeric carbon lies below the plane of the ring in Haworth projection. Trans to CH2OH group in T sugars, right? What about the beta anomer? The hydroxyl group lies above the plane, that is, cis to the CH2OH group in the D sugar or dextrose sugar. In crystalline form, the glucose is purely the alpha D glucopyranose. In aqueous solution at equilibrium, that is approximately 38% at the alpha D glucopyranose, while 62% is beta D glucopyranose, and less than 0.3% as a furanose form. The beta anomer is slightly more stable due to the anomeric effect, right? And reduced steric interactions in the chair form. This distinction is critical because many enzymes like glucokinase, glucose transporters show preference for one anomer over the other. Now, let's talk about the cyclization of glucose. Cyclization occurs when the aldehyde at carbon number 1 reacts intramolecularly with the hydroxyl at carbon number 5 to form the hemiacetal ring, creating a six-membered pyranose ring. This generates a new chiral center at carbon number 1, producing the alpha and beta anomers. In Haworth projection, the ring is depicted as planar for simplicity, with the anomeric OH position below, that is alpha, and above that is beta, the ring plane. Now, let's talk about the mutarotation. Mutarotation is the spontaneous change in the specific optical rotation that occurs when alpha and beta forms of the sugar interconvert in aqueous solution until an equilibrium mixture is reached. For D-glucose, pure alpha D-glucose is at plus 112.2 degrees, right? And equilibrium mixture at 52.7, and the pure, that is beta D glucose, that is almost at the 18.7 degree. This reaction is, the reaction is alpha D glucose, that is, becomes equilibrium at the equilibrium mixture, and that becomes beta D glucose. This interconversion is catalyzed by water and proceeds via the transient open chain aldehyde form. Mutarotation explains why a freshly dissolved solution of crystalline glucose gradually changes its optical rotation. In clinical chemistry, this property is utilized in enzymatic glucose assays that measure total glucose regardless of anomeric form. In aqueous environment, alpha and beta anomers continuously interconvert through the open chain intermediates. The proportion at the equilibrium differs for each sugar. This dynamic equilibrium is biologically significant because it allows a constant supply to the specific anomer required by a particular enzyme or a transporter. Haworth